or today's lab, if you even want to call it that. Nice and easy. All right, let's jump back into chapter 9. The last thing we talked about, if I remember correctly, and usually this saves it for me, this time it didn't, but I think the last thing we talked about was Punnett squares. Does that sound right? I think we ended there like, hey, you guys know all about Punnett squares, so we're not going to say much about it. And then we went into this, or we're about to go into this, genetic alleles and homologous chromosomes. Does this seem correct to everybody?
Right.
All right, thank you. So let's jump into it. Again, because we've already had this lab, which was due this morning or last night, depending on how you look at it, you've already been exposed to this. So a lot of this will be a review. Can anyone tell me what a genotype is? Yes, Thornabar.
The genotype is, it's like, I'm going to use an analogy.
Okay.
It's like, it deals with cells, but it's like if you want to make a product, say like if you want to bake a cake, you got your sperm, you got your egg, and then you have the mix, and it's where they meet in the cell,
Yep.
so to speak. It's not the finished product.
Right. Yeah, you can look at it that way. I like that. So you're talking about basically fertilization, and that's what brings it together. I'm backing up to give you a hint. If we're talking about genotypes, remember, and when you learn this from Monday, on Monday, and you need to remember it, the law of segregation states that gametes, sperm and egg, they have one copy of each allele, right? The rest of your cells, you have two copies. For every gene in your body, you have two copies, right? You have two alleles, one from your mom and one from your dad. And that is a genotype, right? It's what alleles you have. So then what is a phenotype? It's basically a phenotype is the result of the genotype. So as you learned in the lab, like for the dimpled chin, that would be your phenotype. Do you have a dimpled chin or not, right? That's the phenotype. The dimpled chin was dominant, right? That was a dominant trait. So if you have the dimpled chin, that means your genotype was either homozygous dominant or homozygous recessive. But again, the point here, what you need to know for the exam and what you already know, and hopefully you were just too shy to say it because you're afraid of messing up or something, I don't know. But the phenotype, according to your book, is the physical appearance. And because your book says that, that'll suffice for the exam. But I want to point out for your own knowledge, remember, it's not always about physical appearance. Like sickle cell anemia is due to a certain genotype because they have certain alleles. But you can't look at somebody and say, oh, yeah, look, they have sickle cell anemia. Right, so it's not necessarily the physical appearance. The way I would say it, not that it matters because I test out of the book, but the way I would say it is the phenotype is the result of the genotype, right? The genotypes are, hey, these are the alleles you have. And then the phenotype is, what is the result of that? And to remind you what we talked about on Monday, that's what this third bullet point is. This is from the information we talked about on Monday. The experiments that Mendel did with those pea plants, each character that he looked at had the same inheritance pattern. The parental trait disappeared in the F1, one of them, right? Because remember, there's two traits for each character. So for the character of flower color, right, there was two traits. There was either white or blue or purple. And every time he would mix one purebred with another, so the hybrid cross, that next generation would be completely one color, right? It would have that one trait. In the case of the flowers, again, that was a purple flower. The white ones just disappeared. But then again, when he mixed the F1 generation, took one offspring from the F1, mixed it with another offspring from the F1, what he would get in the F2 generation was that other trait would come back. And again, in the case of the flowers, what I mean is that the white flower would be back in that third generation, the F2 generation. And it was always in that ratio, right? You'd have about 75% of one trait and 25% of the other, or 3 to 1, or 3/4 to 1/4. All those things are saying the same thing mathematically, just in different ways. So any questions about that? Hopefully you already know this. Hopefully I'm wasting your time, and in your head you're rolling your eyes like, I know this. We've already talked about this in lab. All right. So now that we've refreshed our memory on this, let's talk about what this means as far as chromosomes are concerned. So we talked about this, but when we talked about this in lab, we weren't necessarily discussing it physically, like how it looks on your chromosome. And that's what we'll talk about here. So the next slide I'm going to show you is going to be a pair of homologous chromosomes. There's nothing to write down on this. This is just an introduction of what I'm about to show you. And then when I show you, you can write it down if you want. But actually, I'll just go ahead and show you. Here we have a pair of homologous chromosomes. And remember, all your cells, other than your sperm or egg cells, right, they are diploid, right? They have one chromosome from each parent. So let's just say hypothetically, this is chromosome number 3. I'm just making that up. Could be anything between number 1 and 23. That's chromosome number 3. This is the one you got from your mom. This is the one you got from your dad. This is the P gene, whatever it controls. This is all hypothetical, right? And in this case, you got a dominant allele from your mom and you got a dominant allele from your dad. So in this case, that would be your genotype. Your genotype is big P, right? Therefore, you are homozygous dominant. So this should be all a refresher from the lab. And you do need to know this, right? You do need to know the difference between homozygous dominant versus homozygous recessive versus heterozygous. So there you go. That's the situation of the P gene. Now let's look at this A gene. From your mom, you got a lowercase a, right? So you got heterozygous. Good grief. I'm sorry. For your mom, you got a recessive allele. From your dad, again, you also got a recessive allele. So in this case, you're still homozygous, right? But in this case, you're homozygous recessive as opposed to homozygous dominant, right? And then here we go on your mom's chromosome. We're going to look at the B gene, whatever it may be. In this case, you got a dominant from your mom and you got a recessive from your dad. Therefore, your genotype is heterozygous. Pretty simple, right? You guys already know this. The only new information here for you guys, well, the information isn't new. It's just the way it's displayed. So this is the first time I've shown you a picture of chromosomes that told you the same thing I've already told you. So now when we talk about homozygous dominant, homozygous recessive, heterozygous, now you can kind of picture it in your mind what it might look like on a chromosome. And of course, this is just an example. And obviously the reason they chose 3 different genes to look at is because they wanted to give you 3 different options, right? They wanted to give you all 3: homozygous dominant, homozygous recessive, and heterozygous. But of course, in real life, a chromosome is going to have many, many, many more genes than those 3. So this is just a simplified example. Also, there's nothing, please don't memorize this. I'm not going to say something like, because some people, I've seen this before, they pay just enough attention to hurt themselves. Like they're zoned out, they're on the phone, or they're playing on the computer or whatever, and they just happen to focus in and see this, and they think, okay, I need to remember that you always get an uppercase P from your mom and an uppercase P from your dad. I mean, no. This is just an example. So don't memorize anything exact on here, right? This is just an illustration of what homozygous dominant, homozygous recessive, and heterozygous is. But it also brings a new word. Actually, I think you may have heard this word in the lab, or at least read it. I didn't use it. And if I remember correctly, I haven't used it yet in this chapter. But loci, gene loci, that is just saying that is the location. So if I asked you, and I wouldn't, but if I were to give you this picture on a lab or on the exam and say, what is the loci for the B gene? And if it was short answer instead of multiple choice, you might say something like, oh yeah, that gene is towards the end of the chromosome. Or if I ask you what is the gene loci for the A chromosome or the A gene, you might say, oh yeah, that thing is located between the P and the A gene. You see what I'm saying? So it's just the location. I remember when I was a freshman, for some reason, this picture really confused me because this thing right here points to this. It says allele, and that says allele. Right? So I'm like, okay, those are alleles. And then this thing points to these things and says loci. So I remember just being confused. I'm like, wait, is it loci or is it an allele? So we're talking about alleles here. All of these things are alleles. And the loci just says this is where they're located. So everybody's clear on that, right? I don't want you to be making the same stupid mistake I made when I was a freshman. And I was afraid to ask. And I just felt so stupid once I figured it out. I'm like, oh yeah, I really overthought that, made it more complicated. So if there's no questions on this, let's talk about Mendels' other law. So we talked about Mendels' law of segregation on Monday. And remember, basically what you need to know for that is the law of segregation says each of your gametes only gets one allele, right? Because as you know from chapter 8, you go through meiosis, right, to make sperm and egg. Therefore, each of those sperm and each of those eggs only have half the number of chromosomes. Therefore, because those genes are on the chromosomes, you only have one allele for each gene. And that brings us to the second one. And you already know this. We talked about this in chapter 8 when we talked about meiosis 1. And we were talking about how it's unique. And I said, you need to know this thing about meiosis 1. And usually I require that you to know that it's from meiosis 1, prophase of meiosis 1. And I messed it up a little bit in the lecture. So I reminded you or told you that you only need to know that it's from the first meiosis, not the second. So here we go. Mendels' law of independent assortment, which you already know. Before we get to that, your book gets a little bit convoluted and swoops in with a different concept that you don't know yet. And that is called a dihybrid cross. Now, let me remind you what we talked about with a monohybrid cross and what you're familiar with. In lab and in lecture, we talked about a monohybrid cross is if you look at something with one trait, like maybe flower color, right? So maybe big P is dominant and it's purple, and maybe little p is recessive and it's white, right? So that's one trait. And then we did Punnett squares, and you can see, all right, well, what happens if we mix a big P and a little p and a big P and a big P, right? We talked about all those different combinations, and we said that was a monohybrid cross because we're looking at one character. And in that example, the one character that we're talking about was P color. Of course, it could be any character. So knowing what a monohybrid cross is, that we're looking at one character and what the ratios are of one character, what do you think a dihybrid cross might be? What is it? Well, no, I'm sorry. Not the ratio. Let me rephrase it. A monohybrid cross looks at the possible outcomes of a mating when we're looking at one trait, one character. So what do you think a dihybrid cross is looking at? There you go. Very simple, right? Dihybrid cross. Now you're mating of parental varieties differing in two characters. So it's not one, it's not two. Now you're not just looking at flower color. Maybe you're just throwing something else in there. Now we're looking at flower color and, I don't know, the color of the pea itself. Not urine, but the peas, the kind you eat. And to remind you, for a monohybrid cross, if you do a Punnett square, and we're talking about here with the typical situation where you had, for the P generation, you had purebreds, right? So you would have, let's see, I'm going to use the letter A. You had homozygous dominant mixed with a homozygous recessive, right? And then the F generation, the first F generation, was always then heterozygous, right? And I'm just reminding you with a monohybrid cross before we talk about the dihybrid cross. So we know that from the F generation, or the F1 generation, excuse me. And this is the part that you needed to know, when you cross the F1 generation, right, heterozygous crossed with another heterozygous, then you always end up with a 3-to-1 ratio, right? And you needed to know that. And I said, your book goes into a lot of detail, but what you need to know for the exam is you need to associate a 3-to-1 ratio with a monohybrid cross. And to remind you what I mean by a 3-to-1 ratio, very quickly. Big A, little a, big A, little a. Again, we're crossing two heterozygotes. That's a big A, big A. That's a big A, little a. It's really hard to draw here. That's another big A, little a, and then that's a little a, little a. So what I mean by a 3-to-1 ratio is, I mean, these three right here all have the dominant phenotype. So 3 of the 4 will have a dominant phenotype. And then this one right here, the one that's homozygous recessive, that is the one that will have the recessive phenotype. All this to say, to remind you from Monday, when you see a 3-to-1 ratio on the exam, you should associate that with a monohybrid cross. Everybody good there? Okay. That being said, now we're looking at the dihybrid cross again. So we're not looking at just one character. We're going to look at 2 characters. Now these 2 are hypotheticals right here, because this is not how it goes. But we're saying if it did happen this way, if these 2 characters were inherited together, like if the gene for pea color, like, again, the pea that you eat, not urine, if the gene for pea color was right there next to the gene for flower color, then they would be inherited together. Because let's say whatever allele you got from your mom for pea color, you would probably get, her allele for flower color too, because they're right next to each other, if it happened that way. And here's what would happen if that were to happen. If that were the case, then the F1 would produce only the same 2 kinds of gametes they received from the parents. And the F2 would still show the 3-to-1 phenotypic ratio. Basically, and let me simplify this for you, all I'm saying here is if these 2 things, if 2 characters were inherited together, then a dihybrid cross would be the same. That's all you have to think about, right? So there's nothing to study here. I'm still leading up to the thing that you need to know. All this bullet point right here is saying is if they were inherited together, if on the chromosome those genes were close together, then nothing would change. All the ratios for a dihybrid cross would be the same as a monohybrid cross. However, they don't do that. They do things independently. And going back to what we're talking about here, which is Mendels Law of independent assortment, right? So knowing that, here's what you need to know. This next bullet point is what you need to know. We're leading up to this. If the 2 characters are sorted independently, which they are, with exceptions, and we'll talk about that later, then the F1 will produce 4 different genotypes. And you definitely don't need to write this part down, because this is just an example, and I'm going to show you on the next page what that example looks like. They're going to produce them equally. And the F2 generation will have 9 genotypes as opposed to 2. Remember, for the monohybrid cross, you had 2 genotypes. You either had the dominant one or the recessive. Phenotypes. And for the genotypes for monohybrid cross, which we're done talking about, remember that was only 3 different genotypes. You're either homozygous dominant, or heterozygous, or homozygous recessive. But here's the number you need to know. I've said a lot because I'm a teacher. If I were a tutor and just telling you how to pass the exam, I would say, okay, associate 3-to-1. Oh, come on. I've already written this in the last slide. It won't let me do it. Associate the 3-to-1 ratio with a monohybrid cross. Associate the 9-to-3 to 3-to-1 ratio with a dihybrid cross. If I was a tutor, that's what I would tell you. Because tutors are mostly there to help you pass and get a good grade, right? I'm here to teach you and have you understand it, but there's the Cliff Notes version. 9-to-3 to 3-to-1 ratio. I see some people taking notes. I'll slow down. Today, when you send me an email for attendance, just like on Monday, if you send it to me before you get the questions, before I get the questions posted, if I get the questions posted, I still haven't even posted them for Monday. Yeah, just make sure you email me before I get the questions posted. And all you have to do is give me the keyword. And the first part of the keyword is it's a compound word that will make no sense on purpose. So the first part of the word is box. So when you email me, say, I'm emailing you for attendance for October 20th. And the keyword is box. And then again, I'm going to give you a second part of that word next. Anyway, I see people are almost done taking notes, which means I'm almost about to show you the slide that illustrates what is on this slide. Does anybody have any questions? Okay. So remember, in this slide that we're looking at right now, we kind of had 2 different main bullet points. We had an if, like a hypothetical, which was not the case, right? That it turned up not being how it works, versus what really happens, which is this bullet point right here, right? That's the one that happens. So the next slide is similar to it. The next slide is going to show you what if it was this. but it's not. It's actually this. So here we go. If the gene for P color and P smoothness, right? So again, those two different characters, what the color of the P is and what the smoothness is, right? So this one, for example, obviously is yellow. This one on the right is obviously green. That's pretty obvious, right? The one on the left is a smooth P. The one on the right is a smooth P, or a rough P. Now, what we're saying in this left hypothetical, which is not a real life example, if these things were inherited together, meaning if you had a big R, you had to have a big Y, right? You never have a big R and a little y because those things are inherited together, right? If that were the case, then you would still end up with a 3-to-1 ratio. Every time you have a yellow P, it would be smooth. Every time you have a smooth P, it would be yellow. Every time you have a wrinkly P, it would be green. And every time you have a green P, it would be wrinkly, right? That would be if they're inherited together. But that's not the case. Almost always, and again, I say almost because we'll talk about the exception, almost always things are not inherited together. So what, in all actuality, what you have is two different genes, each with their own two alleles, and they segregate, assort independently. I don't want to confuse you there. So here we have the heterozygotes. And look, when this thing goes through meiosis, it makes sperm. Those R's are going to split, right? So half of them are going to be big R, and half of them are going to be small r. That's nothing new, right? We already knew that. Except now we're just throwing in the fact that there's a y. And these y's also are going to do their own thing. We're just happen to be putting them on our own picture. And these y's also segregate. You get big y's, right? And you get little y's. That's all we're saying. So it's the same thing. In a sense, if I had more time, maybe I'll make a video some other time and try to share it with you. But to me, it makes it less confusing. Rather than combining this into one gigantic dihybrid cross, to me, it almost makes more sense to have two different monohybrid crosses, like the Punnett squares that you're used to, having two of those instead of one of these gigantic ones. But again, this is a really long discussion to tell you, again, I'm thinking as a tutor here, what you need to know for the exam is you need to recognize this 9 to 3 to 3 to 1 phenotypic ratio. You need to identify that, recognize that, and associate that with a dihybrid cross. And again, I don't know how I would fix this if I was writing this book. But to me, the more important thing that you understand from this is that genes don't travel together, right? There's different genes. The gene, in this case, the gene for P color and the gene for P smoothness have nothing to do with each other. They're on different chromosomes, maybe. They might be on the same chromosome, but if they are, they're probably very far apart. But more likely, one of them's on chromosome number 7 and the other one's on chromosome number 5, or whatever number it may be. The point is they do not go together. To me, that's more important than any of this math stuff, but what do I know? Anyway, are there any questions about this picture? All right. So a dihybrid cross supports the hypothesis, Mendels hypothesis, that each pair of alleles segregates independently of other pairs during gamete formation, meaning the inheritance of one character has no effect on the inheritance of another. So you could be, again, with the P color and the P smoothness thing, it didn't matter. Those things didn't affect each other. And here's an independent work idea. What things are inherited together or seemingly inherited together? And like the first thing I can think of, and I'm going to use the word seemingly because I haven't looked it up. I don't know. You could look it up. When I think about somebody with red hair, there's also other typical traits or characters or traits that they have, right? And we won't get into it because I want you to be able to write it. But when you think about a person with red hair, they typically have a certain skin type, right? And there's also some other things that hopefully you'll find if you look it up for independent work. But those things seem to be inherited together, right? You very rarely see like a red hair with like really dark skin, right? So maybe there is something up there, and you can look that up if you want. This will come back later because there's also there are some situations where one gene controls different characters. And obviously if it's just one gene, then you're going to have multiple characters because and they do travel together because it's the one gene. Anyway, most important thing here, in my opinion, Mendels Law of independent assortment is what we just talked about. Again, these two genes do not travel together, right? They sort independently. And your book uses Labrador retrievers as an example. So being black is dominant, being brown is recessive. Being able to see is dominant, being blind is recessive. However, these things have nothing to do with each other. So because of that, the alleles travel separately. They don't influence each other. So here we go. In this example, and this is just an example, there'll be no questions about Labrador retrievers on the exam. But here we have a black dog that can see. We know its phenotype. It's black. It can see. Therefore, we know a little bit of its genotype, right? And this is important. Not this specific example, but this will come back later. You might get a question where I describe something like this and ask you what the genotype is. Because this thing exhibits the dominant phenotype for both, I know the color and the blindness, we know that at the very least, its genotype is a big B and something and a big N or something, right? Because this something doesn't matter what it is. Because as long as you have one dominant allele, you're going to have the dominant phenotype, right? So we don't know what the other two are. We just know that it has at least one. Now with the blind black dog, again, we know because it's black, that's the dominant phenotype. So we know for its genotype, it must have at least one big B. And again, the second one, we have no idea because it doesn't matter, right? It could be anything. We should use a different example, given the lab that you guys did. Think about like cleft chin and, blue eyes, right? So if somebody has a cleft chin, you wouldn't know what their genotype is, right? Because that's dominant. So they would only have one of the two. If they had blue eyes, you would know that they're homozygous recessive because that's a recessive genotype. Anyway, I'm going to go through this. Those were just some examples. I'm going to come back to that later. The idea here is that based on a phenotype, you should be able to know at least part of the genotype. Anyway, if you were to cross these, here's what you would end up with. If for some reason you had two heterozygote labs, so a black normal vision dog like that, and you cross them, this is the ratio you would expect. You would expect to have about 9 black puppies with vision, 3 black puppies without vision, 3 brown puppies with vision, and 1 blind brown puppy. Again, the only thing you need to know here, you should say, oh yeah, look at that, 9 to 3 to 3 to 1 ratio. We must be dealing with independent assortment. We must be dealing with a monohybrid cross. Excuse me, good grief. Dihybrid cross. And again, this is just an example. And the most important thing you need to recognize here is, again, traits, these characters that have their own genes and their own alleles, they don't influence each other. All right, so any questions about that? We're going to use these dogs again for this next example, and I've been kind of hinting at what we're going to get to here. Using a test cross to determine an unknown genotype. So let me think about it like this. Okay. How many of you have a cleft chin in here? No one in here. Anybody online have a cleft chin? All right. Baumgartner raised his hand. He has a cleft chin. So we know he has a dominant phenotype. What is his genotype? Do we know his full genotype? We do not know his full genotype. There's your answer. All right. All we know is he has a cleft chin. He has the dominant phenotype. Therefore, we know his genotype is this, D something. Because he has the dominant phenotype, we know he has at least one dominant allele. You guys are following me there, right? That's very important. Moving back, that's important. The stuff we've already talked about, that makes that important. So how can we figure out his genotype? This is very unethical because I'm talking about humans, and this is why your book talks about dogs, because that's a little bit less unethical. But to figure out his genotype, here's what we can do. Baumgartner, we know he's got the dominant phenotype. I could pick somebody. Let's see who's online. Actually, I'm just going to make somebody up. I'm going to hook him up with a woman who, let's see, does not have, right? Smooth chin. So if he's hooking up with this woman who has a smooth chin, which is, again, that's the recessive phenotype, what is that woman's genotype? Now remember, she's smooth chin. She has the recessive phenotype.
This recessive.
Yep. So how would you say, what should I write then? If we're dealing with D's, what should I write?
Little D.
Little D? And what else?
Another little D.
Good. So in this experiment, trying to figure out what Baumgartner is, the other Baumgartner all nine, I'm going to force him to mate with somebody. Very ethical, right? Sorry, Michael. Hope you like her. Anyway, so here's the situation. We don't know what his is, right? We're trying to figure out what his genotype is. So there's two different possibilities here. Either A, he's homozygous dominant, or B, he's heterozygous, right? We don't know which. What in the world is he? Now, the female, we know what she is because we can look at her chin. We can look at the hair on her chinny chin chin. Oh, yeah, sorry, Michael. She's also a little bit hairy on her chin. But that has nothing to do with the cleft. So anyway, we know no matter what, she's going to have little d when she produces her eggs, right? Hopefully you guys can see that back there. The next slide I'm going to show you is basically going to be the same thing, except it's with dogs, so it's a little bit more boring. So in this situation, if he was homozygous dominant and he had a bunch of kids with this lady, all their kids, 100% of their children would be heterozygous genotype, meaning that 100% of their kids would have the dominant phenotype. They would have that cleft chin, right? There's no other way. That's the only possibility, unless there was some sort of mutation, which we'll talk about later. So that would be the situation if he was homozygous dominant. Now, if he was heterozygous, you would have a big D, little d here. Here you have a little d, little d. Here you would have another big D, little d. And here you would have a little d, little d. Now, if he had one kid, that's not going to tell us anything, right? Because if he has a kid with a dominant or with the cleft chin, or he has a kid without a cleft chin, that doesn't tell us. That's only one kid. So I'm going to force him, this is how I roll, to have a bunch of kids with this woman, right? And let's say he has, 4 kids. That makes it nice and easy. And 2 of them had the cleft chin and 2 of them didn't. Then we would know that obviously his genotype must be heterozygous. Matter of fact, we would know in this situation, if he were to have kids with this woman, if he ever had a kid that did not have a cleft chin, we would know that he is homozygous recessive. Does that make sense? And it would have to be. That's the only way he can have a kid with her that is homozygous or that has the recessive phenotype. Now, where it gets a little bit trickier, and your book doesn't talk about this, and I'm not going to test you on it. Technically, he could have kids with this woman, over and over and over again. And technically, he could, even in this situation, still have nothing but kids with the dominant phenotype. Does that make sense? This is an important concept here, and I don't know why your book doesn't talk more about it. These are rules of probability. So the rules of probability say if he has 4 kids with this woman, most likely 2 of them will have it, 2 of them won't. But the important thing to recognize here is this is just probability. It is very possible for this heterozygous person to have many kids with this woman and somehow each time end up with the dominant phenotype. In which case, it might look like this situation. Like, well, all the offspring are cleft chin, therefore he must be homozygous dominant. But we don't know that. In other words, what I'm saying is we can prove heterozygote, right? We cannot necessarily prove homozygous dominant. Because no matter what, obviously the more kids he has, the more likely it is, right? If you just keep getting homozygous, keep getting cleft chin over and over and over, the more children he has with that cleft chin, the more statistically probable, right? That sure enough, he does have a homozygous dominant genotype. But that's the point. So now that I've drawn it out, these next slides I'm going to show you, I'm going to go through really quickly. It's just the same thing, except it's from your textbook, and they talk about dogs instead of Baumgartner. So here we go. We call what I just did here a test cross. I'm not going to test you on that word. So don't need to worry about the word test cross. I just showed you what a test cross is. There is a question on your study guide that I might have on the exam, and it says something like, what would you use the test cross to determine? And you would use the test cross to determine the genotype of a parent, right? The genotype of a parent that has a dominant trait. So again, I'm just going to go through this really quickly because this is just reiterating what I've already said. Here's the test cross, but instead of having chins in humans, we're talking about coats in dogs. So this dog is the dominant phenotype, which is black. Therefore, we know its genotype has to be big B something, right? We don't know what this second thing is. It doesn't matter. And we know because this dog is recessive, she has the recessive phenotype. She has to be, or he, whatever, has to be homozygous recessive. So if we mix those two, there's two possible outcomes. There's one in which all the offspring show the dominant phenotype, or there's one where there's a 1-to-1 mix, which mathematically is the same as a 2-to-2 mix. So again, I say the same thing. If you're trying to determine the genotype of this dog that exhibits the dominant phenotype, what you would do is do a test cross, which is where you mate it with something that has the recessive phenotype, and you would look at the offspring. And what we know by looking at the offspring, if we ever get at least even just one offspring with the recessive phenotype, then we know that the other one was the parent was a heterozygote. And the other thing we can say again, if all the offspring show the dominant phenotype, we don't know, but it does point to perhaps that the parent is homozygous dominant. Because again, it is still possible for this situation right here to have nothing but dominant phenotypes. The more you have, the less likely it is, but it is still possible. All right, so any questions about that? Like I said, this is all about probability. These dogs right here, if they were to have two puppies, there is no guarantee they would both, that one would be black and one would be chocolate. It could be two black. It could be two chocolate. It is also possible that they would have, if they were to have 6 puppies, that they would all be black or that they would all be chocolate. Or maybe 5 would be black and 1 would be chocolate. Or maybe 1 would be black and 5 would be chocolate. That would be very unlikely because these are just the rules of probability. Which brings us to this. The rules of probability. To me, this is a very, very important concept, even outside of biology, outside of science, especially in these days of COVID. The rules of probability. Because you're looking at things like, someone says, all right, you have this chance of catching COVID. You have this chance of dying from it if you catch it. And if you wear a mask, it reduces your chance by this. And if you get a vaccine, it reduces your chance by this. And if you social distance, right? So all these things, it's all about probability. Like what is the chance of you doing this based on this? And there's a mathematical equation behind it. I don't even want to use the word equation. It's not an equation. Well, it is, but when you think of the word equation, you usually think of this long, complicated thing. But this is not long and complicated. Okay, so the genetic crosses, which is what we've been talking about, right? All these dihybrid crosses and monohybrid crosses, those are genetic crosses. Those things obey the rules of probability. The same rules that apply when you toss a coin, when you roll a dice, when you draw cards, when you play the lottery, all that stuff, right? So if I were to flip a coin right now, what are the chances that it would land on head? 50%, right? Or you could say 1 and 2, right? It's all the same thing. Well, if I were to roll a dice, what are the chances it would land on the number 2?
1 and 6.
Yeah, 1 and 6. All right. What are the chances it would land on a 4? Yeah, exactly, right? It's just the same. Same thing here. And this brings us to the rule of multiplication. And you need to know this. You will need to apply it at least once. Let me rephrase. You need to know how to use the rule of multiplication. So I'm definitely not going to ask you to define it. But it is the probability of a compound event, or the probability of a compound event is the product of the separate probabilities of the event itself. And that's a little bit confusing, but let's make it easier. Yeah, I'll make it easier with the next slide. So here we go. Rules of multiplication. So we're looking at this female. She's heterozygous. She's going through meiosis to produce some gametes, in this case eggs, right? And those things are going to split, right? Half of her eggs are going to have a big B. Half of her eggs are going to have a little b. So if I were to ask you, like, all right, let's look at somehow we have all of her eggs on a microscope and we can see them if that were possible. And somehow we could even see her alleles if that were possible. And if I were to ask you, looking at this woman's eggs, I want you to close your eyes and pick one of her eggs. What are the chances of you picking an egg that has a big B? 50%, right? Because that's it. Unless something went wrong, 50% of her eggs have a big B, 50% out of her eggs have a little b. Now this dude right here, he doesn't like it, but we cut open his testicles, we gather all of his sperm and put it where we can see it and pick them out, and somehow we can see the alleles. Again, not realistic at all, but let's assume it was. And I were to say, all right, I want you to randomly pick a sperm. What are the chances that it's going to have a little b? 50%, right? Because 50% of his chances of his sperm have a big B, 50% have a little bit, right? So another way to look at that, other than 50%, you could say it's a 1 in 2 chance, right? 1 in 2 chance of getting either one of those things. Now, that's where we apply this to the Punnett square that you've already been doing, but now we're just going to add a little math to it. So what are the chances of, like you already said, what are the chances of choosing perhaps a big B from the female? 1 in 2, right? What are the chances of, I don't know, getting a little B from the male? That'd be this, 1 in 2. So the chances of this happening, then the chances of that happening, that would be 1 in 4, because the rules of probability, multiplication here, right? 1/2 times 1/2 equals 1/4. So the chances of having a big B, little b from this combination is 1 in 4. Does that make sense? So if I were to say, Thornabar, he's going to flip a coin and he's going to roll a dice. What are the chances of him? Let's do this separately. What are the chances of him landing on head? Yeah, half a chance, right? 1 in 2. What are the chances of him rolling a 5? 1 in 6. Thank you. So then if I were to say, instead of him doing this and doing that, and this is not technically correct, if you get into probability, you could argue against this, but I'm trying to simplify things for this example. If I were to say then, what are the chances? You could argue that this is correct. Anyway, if I were to say, instead of these things separately happening, what if I said, what are the chances of him rolling, getting ahead and him rolling a 5 together at the same time? Then the chances would be 1 in 12. That's the rules of probability. If you're saying these two things have to both happen, then it's rules of probability. And your book doesn't get into this, so I'm going to do it very quickly. There's a different thing where you're saying this and this happens. Now, when you say this or this, then you add instead of multiply. For example, what are the chances that there's a couple, they have a kid, everything's normal, we don't know what the sex is. What are the chances that thing's going to be a boy? That couple has a kid. What are the chances that that kid's going to be a boy? Yeah, 1 in 2, right? Because what are the choices, male or female? So yeah, the chance of that couple having a kid is a boy, 1 in 2. Now the second kid, what are the chances? And forget about the first kid. Now we're just talking about the second. What are the chances of that second kid being a boy? And don't overthink it. Forget what I've said earlier. Now this is just a whole completely separate kid. It has nothing to do with the first one, other than it's the same parents. By half again. So if I were to say, what are the chances of them having a boy, and then a boy? Again, like we've learned right here, you could argue that it would be 1/4. But if I were to say, what are the chances that they'll have a boy or a boy? That's like, that's a bad example. Let's just say if it was a boy or a girl, then you would add them, right? So what are the chances that you would have a boy or a girl? Then you would add them. 1/2 plus 1/2 equals 1, meaning there's a 100% chance. 100% chance you're having a boy or a girl. Of course, that doesn't account for genetic mutations and things like that. Anyway, any questions about the rules of probability? Okay. We will use the rules of probability on Friday to talk about family pedigrees, which you might know as your family tree. And instead of just looking at your family tree from an ancestral point of view, like, oh, my great uncle served in the World War I, we're going to look at it from genetic points of view. Like, all right, my great-great-grandfather had this allele, and then so on and so forth, and this is how it's passed along. So the second word, if you are emailing me for attendance, is going to be glass. Remember, those two words have nothing to do with each other. I did that on purpose, but there's the two words that you need to email me for attendance, and the second one is glass. Any questions? All right, I'll be online for lab if you need me at noon. Otherwise, I will see you either on office hours or lecture on Friday. And keep an eye on your emails, because if I get sicker, then we're going to have an online lecture on Friday. So I don't want to make anybody else sick.
What's your